want to do today is share with you a day that changed my life. But I'm going to have to go through quite a bit of background to tell you how I got there. Darren's right. I spent my whole career and eventually retired from the pesticide industry. I was in field research for 25 years, a lot of different capacities, went up through the system, into marketing, and then eventually even launched products. What I thought about was in retirement, we have a brand new citrus industry in South Georgia. Some new developments that have made it possible, and it's collapsing in Florida, so we're growing fresh fruit. And I thought, that would be interesting, and in retirement to grow citrus. And so initially my thought process was, I bet there would be an economic advantage to grow it organically. This is a pesticide person, talking, looking for an economic advantage. And so I started looking into organic. And a friend of mine who actually worked for In-Soil Algae sent me a podcast. The first one he sent me was Christine Jones, then Elaine Ingham, then John Kemp. And this was a year before I started the Grove. I absolutely couldn't believe it. 38 years in the ag industry, and I'd never heard the word regenerative. It's unbelievable. Communication is our biggest challenge. There was no awareness. We didn't talk or weren't aware of soil health, the ecosystem that was alive in the soil. I was developing products that were going to be applied to the soil and that would have wiped out so much of that. So almost immediately, I had a life change. It transformed everything that that I knew about, realizing that there was now research on all of these things, a lot of new information. And I realized we're rediscovering ancient information and technologies, but now we can prove it. We can do all we can evaluate exactly what's going on. So that's my assistant manager at the Grove that's there with me. He really is an innovator. He does a lot of great things. So right from the beginning, so I spent 1 year while I was still working 3 to 4 hours a day listening to podcasts and YouTube videos to learn everything I could get my hands on about regenerative agriculture. I was absolutely transformed by it. And then my goal immediately became, I think I need to be certified if I'm going to do this legitimately. So 3 years in, I'm certified organic and certified regenerative by A Greener World. So what did it mean to me to grow regeneratively? These were my goals, and this is what I did. So my Grove has cover crops year-round. I plant 10 species in the spring and 10 more in the fall. Absolutely tucked to green cover. To me, cover crops are the delivery system. I'm a microbe farmer, and cover crops are the best way for me to deliver microbes to my trees. Once the trees are in the ground, it's a permanent crop. Soil's never going to be disturbed again. So cover crops are a great way to do that. I add microbes. I use compost tea, a lot of microbial inoculants. The soil in my Grove, I bought literally a cotton field. Had been in a cotton peanut rotation for many, many years. And that's where I planted my orange grove. So there's a lot of transformation that needed to take place. It had a lot of things on it. Cotton field basically is bare 6 months of the year and exposed to ultraviolet light, continually tilled, destroying any possible beneficial microbes that might be there. I actually feed the microbes all year long, at least once a month. I mean, they get everything: seaweed, kelp, fish hydrolysate, molasses, long-chain carbons, humic acid, fulvic acid. So it's a continual process. Last week, I literally was irrigating, and I thought, microbes could probably use a little boost. And I had organic cane sugar, and I mixed up a pound to the acre. They love it. So citrus are an interesting tree. They're an evergreen. They don't ever drop their leaves. And so they just continue to grow. Genetically, they could get 5 flushes of leaves a year. So that's a citrus grower's dream. That would be unbelievable to get that much growth continually. Average growers probably get 2 leaf flushes a year. So that was my goal and my dream, to get more. My trees are incredibly healthy. There's not any sign of disease. We have lots of fungal and bacterial diseases on citrus. I attribute that to what's going on, to the microbes in the soil. The growers around me are applying 2, 3, 4 fungicide applications per year, a couple of bactericides, 5, 6 insecticide applications, and then the ground is kept clean. Constantly Roundup or something to have clean ground under the tree. But ultimately, my goal is nutrient-dense fruit. And I guess I had this in the back of my mind, but didn't realize how significant it was going to be, that nutrient density would be really what changed my life. So a professor called me from the University of Florida. I'm not too far from the Florida line. I'm in South Georgia. And he said, there's no other organic growers. And he said, Herb, I've got a grant to evaluate organic citrus versus conventional citrus for nutrient density. Could you do that? I said, yeah. So I proceeded. That was 2 years ago. My very first harvest. There wasn't a huge amount of fruit, but I had page mandarins, and then I had some grapefruit. So I organized and found a conventional grove not far from me, and on the same day, I went and picked my mandarins and mandarins from the conventional grove, cut them and juiced them and froze them. And then 3 weeks later, I did the same with the grapefruit when the grapefruit were ready. Went to a different grove. And these growers, they're excellent growers. I even bought my trees from the grower that has that fruit, has a nursery. And so froze it, labeled everything, and waited for the unit. I lined up the laboratory to analyze it, which is, at that time, it was horrendously expensive. Now we have audacious, thanks heavens, that it's something that actually a grower can afford. So I waited and waited, 2 months, 3 months for the university to call, say, yeah, go ahead and send the samples off. When he called, he said, I'm afraid the funding for the grant has fallen through. And I thought, oh, great, I've got a freezer full of frozen juice samples. I thought, this is not going to go over well at home. And my dear wife, after about a month or two, seeing me struggle over it, she said, if you're going to do this, you might as well just spend the money and go get the samples analyzed. So I did. So what happened, this was probably the start of the day that changed my life. The day that changed my life was the day that I got the results back from the laboratory. I knew I could get a 50% bump in nutrition, and I really hoped for 100%, but 800%. Had absolutely no idea. I was floored. This is a consolidation of, they analyzed 26 different things. 9 flavonoids, 11 carotenoids, and 5 or 6 vitamins. And it's complex data. There was a lot of it, but when you condense it and try to analyze across it all, that was the difference. My fruit was 8 times more nutrient dense than conventionally grown fruit. So what I'd like to share is some of the details of that. So these are some of the flavonoids, and coming after sherry is perfect. Flavor is nutrition. Where does flavor come from? From flavonoids. So these are the world's best kept secrets. I mean, flavonoids. Citrus has 90 different flavonoids. 90. Each one of them, almost all of them, are antioxidants, anti-inflammatory, and then they all have a unique role, some health implication in the human body. And so I get this data and start going through it and analyzing it. Across the bottom, I'll just read them to you. Didyamine, hesperidine, nerintin, and then total flavonoids. These 3 here, they're completely missing in the conventional fruit. To be honest, that's scary to think that we have fruit, any produce, that's going to end up in our grocery stores that we're going to eat, and there's components that we need for nutrition. Well, they're not at very high levels anyway, but for some of them, they're completely missing. All 3 of these, was interesting, were involved with brain function. I mean, think of that. So the ones that were missing were ones that were very significant, that would have been important for us. So I tried to think through, okay, this data is complicated. I built charts, which is what I always have done, and showed it to some people and to my family, and they said, no, that doesn't make any sense. So they came up with this new communication system. 1 orange stands for the level in a conventional fruit. And I've got some of these charts that I brought with me if anybody'd like one. It's a long ways away to read that. And then my fruit had this increment, this much more. There were actually 4 of them, of the flavonoids that were missing. And then I correlated for each one what the health benefit was. Having AI now, it's an incredible tool to go into ChatGPT and go through all these things and find out what are the benefits, what exactly do they do. This one that is so astronomical, hesperidine, when you dig into it, it's circulation, blood circulation. It has a direct impact on cognition, brain function, and memory. And my fruit was 39 times higher. So this is just about a memory bomb. So this was a way that I chose to try to communicate it, and I think getting our message out is so important. So I've made it into a little chart, and I include it in a box of fruit when people get it. I want to be able to see what it is that they're getting, and I know I'm talking about citrus, but this applies to all crops. It applies to the vegetables that we saw yesterday. It applies to cereal grains that are raised regeneratively, and it applies to the beef, to the to all the meat that you all are raising and selling. This is just an example, but nutrient density is there. And it's something that, just like I said, that as somebody in big ag, in that industry, industrial ag, this is not being discussed. There was no information, not even a hint that somebody. I saw YouTube this week, and it was one of the famous ag guys in the Midwest, ag PhD. Everybody follows him and watches him, and he literally said, organic food is dangerous to you. I just couldn't believe it. That's the saddest message that, unfortunately, is being perpetuated. So a little bit deeper dive. Again, this is pretty tough, but this was my way of evaluating, trying to categorize what all of these nutrients in my fruit did. So across the top is anti-inflammatory, antioxidant, anti-cancer, brain function, eye health, heart health, and skin health. So you can see immediately there's kind of an interesting comparison between flavonoids and carotenoids. The flavonoids, absolute anti-inflammatory, which is the basis of so many chronic diseases. Both have excellent characteristics for antioxidants, anti-cancer. Most of them. And then when you get into the carotenoids, a big boost in eye health that we all know about, beta-carotene. But some of the most interesting ones, on the top left there is naringin. And so one day I was going through the grove. If a fruit is scarred or knocked off a tree, I cut it and eat it. And ate a grapefruit while I was out in the grove, and you've probably had this sensation, it makes your lips numb. Mine makes your lips really numb. And I was working for the day, and 4 hours later, my mouth was still numb. Now, it's not a bad numb. There's a little hint of flavor, and it's kind of a tingle. But 4 hours later, my mouth was really numb. So I'm in the grove, and I thought, well, that is from the grapefruit. There's no doubt. So I get out the phone, ChatGPT, look up. Guess what it is? It's naringin, one of the flavonoids that I had analyzed, and I'm thinking this is an incredible effect. So I have done a deep dive into that flavonoid in particular. It's an incredible anti-cancer product. It actually inhibits tumor formation. It reverses tumor formation. It's being evaluated as a potential pharmaceutical, naringin. This thing that I'm walking around the grove with my numb mouth. And I'm thinking, that's great. I don't care how much it hurts. Think of the boost that I'm getting. If it lasts this long, my body is getting supercharged with this product. Not only is it being evaluated by the pharmaceutical industry, it's been documented as the effect that it has on 14 different forms of cancer. Why are these things not even being talked about? That's incredible to me. So, in our fruit, and there are 90 different flavonoids. Now, this may be the downside of growing regeneratively. As Darren mentioned, I did research a lot of my career, 25 years. So in my own grove, that's continually what I do. There's a lot of practices that nobody can give me advice. I don't know. There's no documentation, so I do research plots. And I don't mess around. I do replicated plots. I take data. I spent the first 3 years measuring trees down on my hands and knees, seeing how quickly they grew. I research everything. Compost, the treatments, in-soil algae. I've evaluated it several times. All the organic insecticides, which there's no information about what's going to control the leaf miner that I've got. But when you use synthetic fertilizer, this is what you get. You get a bigger tree. On the left is my regenerative. This was a big trial. I did was 135 trees. A row is a plot, and I had 3 treatments, and I replicated it 3 times. So it ends up being a lot of trees and a lot of data. In 2 years' time, they were twice as big, and by the 3rd year, they were 3 times as big. So, but there's a reality to it. It's a sacrifice that I felt comfortable making because I knew the quality of the fruit that I was going to produce. Think about it. Everybody knows the nitrogen stimulates growth. That's why growers use it, big and tall, increase yield. But if that's literally creating cell expansion and there's not the horsepower, there's not the physiological changes in that tree to fill it up, those are hollow cells, hollow trees. And what I've shown in my analysis is hollow fruit. That's basically what it comes down to in the end. So there's a lot of changes in my grove. The most interesting is the soil. Literally, for people that are not farmers here, this is an exciting thing. This is one of my favorite tools. It's called a penetrometer. It's a 3-foot-long rod with a pressure gauge built inside of it. And so you push down on it with 200 pounds of pressure, and you see how far it goes in the soil. Well, in between the rows, every tree that I have, the way we grow them, there's a microjet sprinkler under every tree, and I can put everything through there, all my nutrition. I can inoculate microbes. I can feed the microbes through the tree, and it goes a 6-foot radius around the tree. So I can go a few feet outside of that and put the penetrometer in, and it goes in 3 inches. Even though there's cover crops there, that's all it'll do. And then I come 3 feet over next to a tree, and it sinks just about to the handles, 30 inches, 36 inches. The ground is actually spongy, and that's got tree roots, cover crop roots, but what it's really got are microbes, the proliferation of microbes that I'm constantly guarding. In the summertime, I watch the temperature. I have temperature and moisture gauges all through my grove. I don't let the temperature of the soil get above 85 degrees because it can kill microbes. So that's when I irrigate. I irrigate to save the microbes. At least every month, I'm feeding the microbes. And they're returning that in the incredible fruit. And the organic matter in my soil has increased from 1.1% to 2.2%. They don't sound like big numbers, but in South Georgia, sandy soil, nobody has got 2% organic matter. The organic matter is what fuels the system, keeps the microbes alive, keeps everything going. One of the most interesting projects that I've done is to do DNA analysis of my soil. I didn't realize what a nerd I am, but I guess that kind of proves it. So before I ever planted my grove, I applied for a grant with Biomakers, which is a company that does DNA analysis. And they have supported that trial with the regenerative versus conventional for 4 years. And so this was This is one of the visuals that you get. They analyze bacteria, archaea, all the fungal species. They've got a catalog of 24 million soil microbial species that they've identified. And so this was a pre-sample before I ever planted my trees. So I knew what the baseline was, how much, and I was shocked. There was 600 species in this lackluster soil. So just after 7 months, I looked at, thanks. I looked at both the bacteria and the fungi. So this was the pre-treatment. Here was a conventional treatment. The graphs are the quantity of fungal species. The conventional actually went down. And I had a middle treatment of indigenous, organic with no microbes, and then this with me added the microbes. So this big increase, a doubling in the quantity, but the number of species across the top went from 268 to 507. That was incredible. In 7 months, that was my first season. And then after 3 years, I continued to look at this. On year 3, again, the fungal population was now dominated by an absolutely worthless dwarf puffball. 89% of all the fungal activity, whereas mine still had those 500 species with a great diversity. That's what we're always trying to get, diversity, whether it's bacterial or fungal. That's what gives the soil strength and resilience. So it's been truly interesting to be able to track it now with these scientific tools that we have and to know everything that's going on in our soils. So I'd just like to end it there if we got time for a question or two.
Thank you, Herb. That was amazing. I'm sure there are lots of questions. Who's got one to kick us off? All right.
Hello. My husband is also a tree farmer. We farm pecans here in Oklahoma. And we moved from a conventional model to a more regenerative model starting about 5 years ago. And our journey has not been a smooth road. It's been pretty rocky. It's good, but there's been a lot, like fungal issues are huge in Oklahoma. We've reduced our fungicide by 33%, and then we had the wettest spring we've ever had, and we had a ton of fungal pressure. We are applying a lot of what feels like a lot of biological products. Is very expensive, but definitely not every single month. So my question for you would be, how can you affordably feed your soil microbes every month? Like, do you have any recommendations about that?
I'm having some of the same challenges. I mean, I hear vegetable people talk about, there's not much cost difference. For me, there has been. A lot of it's labor, but the products that we're needing to use. Some of the things that to support soil microbes, literally, sugar is something. Organic molasses, some things that are not so expensive. But I really sympathize. Yeah, I think, there are organic products. I think fungal problems on the leaf until the tree developed, it's called this phyllosphere, develops this population that's on the leaf that's going to protect you. But brewing compost tea is probably one of the best ways to achieve that. Brew compost tea, and I've got kind of a homemade brewer, and then one that's more complicated, and then put them out through the sprayer. You can spray them on the leaf, put it on the soil. Those are great contributions to try to develop the microbes. But I agree, it's not a simple task. I've run into lots of challenges.
We've got another question over here.
Maybe you don't have time, but curious with that 8X that you distilled your nutrient density testing to the 8x comparison to conventional, I mean, I know there's a lot of different parameters that you're looking at, probably a lot of variability when you compare conventional to the regenerative, but what was your approach to distill it down to that 8x statement?
Sure. So basically, I went through, it's 102 data points. When I had 2 varieties, and they were looking at 26 different things. So the data I got back was page mandarin, conventional regenerative. And so I calculated what is the increment, what's the increase for Naringen. It's a 1.2 or 5-fold or 8-fold. And then I did that for every variety, for all the grapefruit, for all 26 of the things that they analyze, and then I did it for all the page mandarins and all the grapefruit. So it's paired comparisons. It's not actually replicated, which I'm going to try to do this year. I'm working with Kiss the Ground, had some small grants, and that's going to help pay for some of that. So this year we're going to replicate it, and then we're going to match it with soil DNA evaluations also. From the trees that I take the fruit sample from, we're going to find out what the soil population is. So, and then the dream is to try to find a link between the two, if there's causal. So, yeah, all that 8x is a summary across all 26 of those things across 2 varieties. So there was a lot of variability, exactly like you said, and it was hard to factor in the ones that were missing if there's no flavonoid. So I gave them the benefit of the doubt, and I gave them the lowest level to be able to have to figure out if I was 5x or 10x. If there's 0, there's not a way to calculate. So it was complicated. So that's actually a conservative number.
Are looking to purchase microbial inoculants or soil amendments, what would you say would be kind of your top 3 or 4 things that you look for in those products?
Sure. I really believe that there's a freshness aspect to microbes. I bought some mycorrhizal fungi that were out of a barrel in the back of the warehouse, and I didn't see any impact from them. So I've actually tracked down a mycorrhizal fungi guy who develops an orchard product. And I asked him, when is it fresh? When do you make it? And I get the first batch that he produces. I get a lot of products from Tanio, and they're very dependable, and they list what microbes are in their product. That's pretty amazing. And then having analyzed them, there's even more in there that may not be listed. So brewing your own is another good way to do it. I take compost that I know is active, and I brew a small batch, 10 gallons, and then I put it under a microscope and see how much life is in there. And if it's great, you can scale it up to a tote and do 250 gallons of it. And that goes a long ways.
Hello.
Yeah, hey.
What cover crop mixes do you use for your orchard floor, and then do you envision using cover crops indefinitely, or do you think you're going to transition to some other kind of cover?
I wish that they would become perennial and that I wouldn't have to keep reseeding them every year. That would be the dream. In the winter, I have great luck. I seeded 2 weeks ago. To me, cover crops, there's 2 great functions. They feed the microbes. 40-50% of everything they produce goes down to feed microbes. But even better than that, to me, I use them as carriers for my inoculant. So my trees are permanent. They're in the ground. I can't get to their root system, but the cover crops can. So I heavily inoculate the cover crops before I put them out. I put mycorrhizal fungi inoculant and then 2 different bacterial inoculants. I've even mixed dried molasses cattle feed with it to feed the microbes as the cover crops are going out. But I try to do a complete mix, which all these company green cover can put together a great mix that fits your area. But it's about a third legumes, 2 grasses, daikon radish, 3 different brassicas, all in there for the winter time, and then Austrian winter pea, hairy vetch. That's a great one for the south. In the summertime, we have better luck with sun hemp, buckwheat, and then all the grass species do well. And then clovers all year round. We have some summer clovers that I can use in South Georgia, and then I use 3 different species of clover in the wintertime.
Thank
Yes,
you.
go ahead.
How can we utilize some of these practices on the soil, feeding the microbes when we're using dryland farming and pasture management?
I'm sorry, one more time. I couldn't quite catch it.
On your on your soil, on feeding your soil microbes, how do we utilize those practices in dryland farming and pasture management?
Even though I have irrigation, the irrigation is just around the tree, so I put out cover crops. I wait till I have a great weather forecast of 2 or 3 days of rain. So I want to put them out when they're going to get a rain right behind them. It's a challenge, especially in the summertime. We have real high temperatures, and if I don't get them out early enough, I've gone in the wintertime, I just spread them with a fertilizer spreader, but in the summertime, I've had to use a no-till drill to put them in, and I get great germination that way, as long as there's some soil moisture. But if there's not enough, those seedlings don't do well and survive. So a lot of them I lose in the summertime. The grasses push right on through and do well, but some of the broadleaf stuff has a real tough time. Yeah, it's synchronizing it with the rain, and that's what I have to do too.
Herb, we have a quick question over here. We know you're in South Georgia. What growing zone are you in?
Growing zone?
Yeah.
It's 8B.
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 Okay. Thank you.
Yeah, I'm right at the northern limits for citrus. We have a new cold hardy rootstocks, and we have this new irrigation methods by putting a sprinkler. I run the irrigation. I'm out there all night long when it's going to freeze, checking those nozzles to make sure they haven't frozen. Those 2 things keep the trees alive during a freeze. That's why the citrus production has kind of worked its way north a little bit. So.
We have time for one more question, and my mic is maybe going out, or it's just me. Any other questions? All right, Patrick, I'm coming to you.
You've put a lot of work into describing the quality of your fruit. What do you do with that, or who buys your fruit now?
I'm only into my 2nd season. Last year was my 1st season. I sold 2/3 of my crop retail. I sell it all online, and 1/3 was wholesale to farm markets, people that have probably their own store, that are market gardeners, and I've sold it to them, and then they resell it. But we've had to. It's customer acquisition is tough. So we've done a lot of marketing, and it's been really expensive to find. There are people out there, but we shipped to 36 states last year. My first crop. So Instagram or Facebook advertising, and you have to pay for it to get the exposure. Even LinkedIn, where you can talk about more scientific stuff, and people come and find the site. So you got to develop a fantastic website. You got to have killer social media. You got to pay for advertising, Google ads. You got to have branding, which all 4 of those things, that's marketing. All 4 of them are expensive. But that's what it took. We felt that we were so specialized that that's what it was going to take to find the people that really were seeking nutrient-dense fruit.